Hi friends, hope you are fine. At the end of this discussion, you will be having a thorough understanding on the difference between Eubacteria and Archaebacteria within 5 to 10 minutes. Let's begin with the similarities. Both Eubacteria and Archaebacteria are single-celled prokaryotic microorganisms. Prokaryotic means without a true nucleus as you see here, and also without membrane-bound organelles like mitochondria, Golgi apparatus, etc. Now let us see the difference. Let's begin with the definition. Eubacteria are called true bacteria. Eu means true. They are large group of diverse bacteria with rigid cell wall, with flagella in the case of motile bacteria as you see here. DNA is single circular chromosome that is distinct from Archaebacteria. They are of different shapes as you see, rod shaped, maybe sometimes coccus. As you see here, if it is in chain, we call it as a streptococcus. In the case of Archaebacteria, archaea are called ancient bacteria. It is considered to be an ancient form of life that evolved separately from bacteria and blue-green algae with distinct characteristics. Archaebacteria is a mysterious group of bacteria that shows characteristics of both Eubacteria and also eukaryotes. In some recent classification system, because of the distinctiveness of this Archaebacteria, they are even considered as a kingdom. They also appear in different shapes under microscope as you see here. Difference number 2 regarding the occurrence. Eubacteria are found almost everywhere on earth. In freshwater, saltwater, human guts, as parasites, as saprophytes, and different modes of life. Whereas Archaebacteria mostly grows in extremely harsh environments, therefore also called as extremophiles. In highly acidic condition, in high temperature, high salinity, some are methanogenic. In muds and marshes, they mostly prefer to grow in harsh environments, even in Dead Sea. But recent discoveries suggest that they are present to live everywhere, from ocean floor to the cow gut. They are present everywhere, in soil, in ocean, and also inside organisms, just like Eubacteria. Difference number 3 regarding the cell wall. The cell wall of Eubacteria is made up of peptidoglycan. As you see, it is divided into gram-positive and gram-negative bacteria. As you see, the cell wall is made up of peptidoglycan. In the case of gram-negative bacteria, there is an outer membrane that is made up of lipopolysaccharide. This peptidoglycan is made up of N-acetyl glucosamine and N-acetyl muramic acid that is joined by short peptide bridges or short chain of amino acids. As you see, these are D amino acids, D-glutamine, lysine, alanine, etc. Whereas Archaebacterial cell wall is quite distinct. It is composed of various polysaccharides and glycoconjugates. In some Archaebacteria, there is pseudopeptidoglycan. It is also called as pseudomurine. Instead of N-acetyl muramic acid, it contains N-acetyl allosaminoronic acid or NAT that is linked by peptide bridges or short peptide chains. They are called as pseudomurine or pseudopeptidoglycan, as in the case of Methanobacterium, Methanobrevibacter, etc. So they are having a distinct cell wall composition. Then the next major difference is in the plasma membrane. In the case of Eubacteria, as you see, this is a phospholipid bilayer. Then this bilayer, the glycerol is, the chirality is D-glycerol. As you see the orientation, this is red color is a glycerol. So the chirality is D-glycerol. This glycerol is connected to this fatty acid. This is a fatty acid chain by ester linkage. As you see this bonding in yellow color, this is ester linkage. And side chains are unbranched. As you see, it is made up of fatty acids. So this is a plasma membrane of Eubacteria. Whereas in Archaebacteria, it is often a monolayer. Sometimes, rarely, it can be a bilayer. Then the second difference is the glycerol is L-glycerol. As you see the orientation here, it is D-glycerol. Here the glycerol, this carbon or this CH2 is oriented to this side, that is L-glycerol. Then the bonding, this glycerol is attached to this side chain by ether linkage. The bonding is CO bond as you see here. This is the ether linkage. Then the third difference is the side chains are made up of isoprene units. They are not made up of fatty acids. They are made up of isoprene units, and also the side chains are branched. So Archaebacteria has a unique plasma membrane composition that is quite different from Eubacteria. Difference number 5 regarding ribosome. In the case of both Eubacteria and Archaebacteria, both with 70S ribosome, made up of small subunit of 30S and large subunit of 50S, forming a complete 70S ribosome. But archeal ribosomes have a different shape than eubacterial ribosome with different protein composition, making it unique. Therefore, archeal ribosomes are resistant to antibiotics that inhibit ribosomal function in Eubacteria. Difference number 6 regarding flagellum. Eubacteria, the flagellum is made up of a protein called flagellin. The rotation is proton driven. Whereas in Archaebacteria, it is having a distinctive flagellum. Now it is renamed as archeallum. It is not made up of flagellin. It is made up of pseudopyelin. It has close similarity with the protein that makes pilus in the case of Eubacteria, the structure that forms during sexual conjugation in the case of Eubacteria. And the rotation is ATP driven, not proton driven. So the flagellum is also unique in the case of Archaebacteria, now renamed as archeallum. Difference number 7, RNA polymerase. In Eubacteria, there is a single RNA polymerase that is made up of 5 subunits. As you see, 1, 2, 3, 4, 5. So this is the structure. Whereas in Archaebacteria, there is a single RNA polymerase that is made up of approximately 11 to 13 subunits. That depends on the species. And it is more similar to eukaryotic RNA polymerase 2. So strong affinity towards eukaryotes in the case of RNA polymerase in the case of archaea. Difference number 8 regarding antibiotic sensitivity. Eubacteria is sensitive to antibiotics. We know this penicillin. Most Eubacteria are sensitive to penicillin as this penicillin inhibits the peptidoglycan synthesis or cell wall synthesis. Whereas, as we said, archaebacterial cell wall is not made up of peptidoglycan. Therefore, Archaebacteria is insensitive to many antibiotics. But it is susceptible to some other compounds or protein synthesis inhibitors like fusidic acid and imidazole derivatives. Difference number 9. Photosynthesis and methanogenesis. In Eubacteria, cyanobacteria are photosynthetic eubacteria with the ability to evolve oxygen just like plants, green plants. Bacteriochlorophyll is involved in the process. Methanogenesis is absent in Eubacteria. Whereas in the case of Archaebacteria, oxygen evolving photosynthesis is absent. In the case of Halobacterium, there is photosynthesis and bacteriorhodopsin is involved in photosynthesis. There are methanogens in the case of Archaebacteria or methane producing microbes, and that produce methane or converts carbon dioxide to methane in their normal metabolism, or methane is produced as a metabolic byproduct. So methanogenesis is common in Archaebacteria. Other differences include the initiator tRNA of protein synthesis in Eubacteria is formulated, or it is tRNA F-met, formulated methionine. Then capsules are present, an outer covering that is made up of polysaccharides are present in most of Eubacteria. Hami and cannulae are absent in Eubacteria. Whereas in the case of Archaebacteria, this initiator tRNA is tRNA methionine without formylation, just like eukaryotic initiator tRNA. Capsules are rare or absent. There are 2 specific structures that is unique to archaea. The first one is Hami, this hook-like projections arising from the cell surface that helps in cell-to-cell contact and also attachment to other surfaces in the case of Archaebacteria. Then the second structure is called as cannulae. So these are hollow tube-like structures that helps in cell-to-cell attachment after cell division, that helps in the formation of a network of cells. So these 2 structures, cannulae and Hami, are unique to Archaebacteria. And finally, let us conclude with some examples. Eubacteria examples include E. coli, Streptococcus pyogenes, Lactobacillus acidophilus, that is in milk, then cyanobacterium or blue-green algae, photosynthetic eubacteria. Then archaebacteria includes Halobacterium salinarum, then Salinibacter rubor, and Methanococcus janus G. So these are some of the examples. Hope you are clear with the difference between Eubacteria and Archaebacteria. If you know anything additional, please add that in the comments. If you find this video useful, please subscribe and support this channel. Take care, stay blessed. Thank you so much. You are with biologyexams4u.com.